Hello everyone, and I'm Li from China, the second year student of MULTI-CELLULAR, and I did my thesis in CCMAR, the University of Algarve, Portugal. So today I'm going to present my work. How did I construct the evolutionary history of Palmophyllales? My work is mostly about phylogenetic study, so it's mobile informatics. So first off, I would like to give a bit background information of a variety plantae, the green plant kingdom. This kingdom contains two clades, the Stroptophyta and Chlorophyta. Stroptophyta evolved from seawater to freshwater and give rise to all the land plants today. And Chlorophyta contain co-Chlorophyta, and their early divergence prasinophyta groups. Co-Chlorophyta contain more than 90% species in this group, while only a few species were found in prasinophyta groups. They were named from clade 1 to clade 6 and more, just because in the beginning people couldn't identify those species. And in the study of Lillard 2016, they used Chloroplast genome, indicated that the prasinophyta clade 6, which called Prasinococcales today, and our study group, Palmophyllales, is the earliest diverging of the entire Chlorophyta. So they named these two groups as a new class, Palmophyllophyceae. So this new class, is the sister of the rest of entire Chlorophyta. So more information about this new class. Of course, our study group, Palmophyllales, contains two genus, Palmophyllale and Vedigelas. They were found from in a very low light deep sea environment from the sea surface to about 200 meters. And other Prasinococcales contain Prasinoderma and Prasinococcales. As you can see from the picture of the specimen, the biggest difference between Prasinococcales and the Palmophyllales is Palmophyllales, our study group, is macroscopic algae, while Prasinococcales is unicellular. So same in the study of Lillard. This is the result tree they have based on the Chloroplast genome. They resolved that this new class is the most basal branch of the Chlorophyta. But here, they resolved that Prasinococcales is a paraphyletic group, which means Prasinoderma is the sister of the group of Prasinococcales and Palmophyllales, but this result doesn't get enough support here, as well as some other Prasinophyta groups, like they don't have enough support in their branch. So what my thesis did is we use more advanced, more complex, and more specific model to the data here, and we try to resolve the internal relationship of Palmophyllophyceae, and best idea to give the support to the relationship and also for the rest Prasinophyta groups. So first of all, I set up my data set by the Chloroplast genome. But why I use Chloroplast? Chloroplast has their own genome, and their gene is much simple than the nuclear genes. So in plants phylogenetics, it's very ideal data to study. And in the study of Lillard, they use 59 taxa within 71 genes. But for my thesis, because I want to use more complex model, which means longer time analysis but I only have half a year. So I have to reduce the number of taxa to finish my series. So in the end, I used 16 prasinophytas and 6 chlorophytas, co-chlorophytas, as well as 6 Chlorophyta as out group. So in total, 28. And 4 of them is new, added from NCBI. So 102 genes were retrieved from 28 taxa, but I only kept 74, which present in the majority taxa. In 74, 5 genes were removed due to their low quality alignment, and some of them are not coding genes. And after we built the gene tree for each genes, we delete RPS4 and ACCD genes, because they have wrong position of outgroups in their result tree. So after concatenate the 67 genes in 28 taxa, I had my own nucleotide data set, and as well as the protein data set after being translated. So here is our first analysis. We know my data set is coding gene data set, which contains a bunch of synonymous substitution. And this substitution won't influence the sequence of amino acids. So if we involve them in the analysis, well, it will give misleading to the result. And most synonymous substitution happened in the third co-position. So the third co-position evolved much faster than the first and second one. The typical method is just simply remove the third co-position in the data set, but it has 2 problems. First, it's still losing the information in the third co-position of the non-synonymous substitution, and also induce the synonymous substitution in the first and second co-position. So here is an alternative method, the degenerate-codon recording, which use same characters to replace all the synonymous substitution. So for compare, we use 2 different methods, record our data set, nucleotide data set, and analysis by maximum likelihood under the GTR gamma-4 model. So here is the first result. We can see the left side is the typical method, and the right side is the degenerate method, the new method. And a short explain. Different clade was colored differently. So our our study class, Palmophyllales, was colored in red. And we can see the biggest difference between 2 data sets is their typology is different, but the right side result from the new method is closer, is more similar to the previous study, and it got more support in the expect position here compared with this removing third co-position method. So after the nucleotide analysis, we start with our protein dataset. But protein analysis is much more complex than the DNA because it has much more parameters in their substitution model. You can see the numbers here. So the typical method for scientists is use an empirical model, a fixed mixed maxtra. We have a bunch of computed model. CPREV and GCPREV is particular for Chloroplast, which means CP. But I want the model is more specific for the prasinophyte group. So I used FastMG and to produce my custom model, and I call it prasREV, prasinophyte reversible model. For compare, the per-computed models and custom models was analyzed by the maximum likelihood method. So here is the result, same as the previous one. Different clades were colored differently. And here, you can see this is a star mark, which means got fully support in these nodes. And first of all, we compare the maximum likelihood of two trees. The left is prasREV and the right is CPREV. prasREV, our custom model, has much higher likelihood than the CPREV, which means my specific prasREV model fits our data better than the CPREV or other models. And also here, in the tree of prasREV, this point in the Palmophyllophyceae, it has a new typology, which is different from any of previous study, that they placed Palmophyllales as the sister of the prasinococcales. And this is a new result. And for further study, most of phylogenetic study is based on homogeneous model, which they assume that the composition of the amino acid or nucleotide is stationary. But in reality, because of the selective pressure or random mutation, the composition of the sequence is not always stationary. So I use a non-stationary model called NDCH2. The full name is here. This model allowed different composition among different taxa. And this model is based on Bayesian analysis. And here we use P4 platform. So here is the result of our NDCH2 model with homogeneous model compare. The right hand is NDCH2 and the left hand is homogeneous. And first as well compare the likelihood, and NDCH2 model fit our data better than the homogeneous model. And also we can see here the backbone of the tree. Compare with this point, NDCH2 solved most of clade relationship and gave the support. So the second part to zoom in our study class, in the NDCH2 model, it happened the same typology as the maximum likelihood of prasREV that place Palmophyllales as the sister of prasinococcales. So this is the different typology between the homogeneous model and NDCH2 model in this class. It probably because of the composition effect. Because in our analysis, we did the composition test, and the result shows our data set is heterogeneous. So in conclusion and the discussion, we build up two models, the prasREV models, and in maximum likelihood, it fits our data better than all of other models. And also because our data is heterogeneous, so the non-stationary model, NDCH2 fit our data better, and also significantly improve the support of the clade, of the relationship of the clades, prasinophyta clades. And also the most important result that we use prasREV model and NDCH2 model resolved and give support of the relationship of Palmophyllophyceae class that multicellular Palmophyllales is a sister of unicellular prasinococcales, which is very different with previous study. So that's what I did out in my thesis, and thanks the CCMR and MBRC give the opportunities to do the thesis, and also my supervisor Philip and Simon, thanks them, and also thanks my colleagues and my family and my friends.